Oh, now. All right. I have to use this thing, so bear with me. Um, Good morning, everybody, and welcome to another seminar. Now, uh, continuing our efforts to bring colleagues from the consortium schools that the CLAB represents, we have Dr. Chuck Amsler with us today from UAB. And Chuck was trained as a phycologist, but you will see during his talk today that ever since has done a lot of work looking at the dynamics and trophic ecology of benthic communities in Antarctica, particularly looking at all this chemical warfare going on between plants and grazers and how plants defend themselves against grazers, and a lot of good papers on those topics, quantity and quality wise. Just a little reminder here that the idea for this series of seminars within our bigger series is to bring people from our consortium schools and to get to know each other better and maybe look for potential collaborations. So as Chuck is giving us his talk, just look for potential avenues to collaborate. I see a lot of synergy between what he does and what we do here at the CLAB. So again, we represent a lot of schools, and it's in our best interest to take advantage of all this intellectual capital we have and maybe think about potential collaboration on the road, okay? Chuck has some time to meet with folks this afternoon, so if you're interested in talking to him, step out, talk to me, and we'll make sure that you connect. Chuck, thank you for coming.
Thank you. So I hadn't thought about it till a few minutes ago, but the last time I was here giving a talk, Rich Aronson had me down. It was probably 98 or 99, and it was here that I found out that we'd gotten the first NSF award of the stuff I'm going to be talking about today. So the CLAB has a very prominent place in my mind about this work. So the work that we do in my lab is primarily things that can be described as chemical ecology, which I like to describe as the study of chemically mediated interactions between organisms or between organisms and their environment. And these interactions tend to fall into one of three categories. The first is chemical communication, and that can be intentional forms of communication, like an insect secreting a pheromone to attract a mate, or it can be unintentional forms of communication, like a predator picking up on the scent of a prey to be able to track and, for the predator's perspective, hopefully consume it. A second area is environmental sensing. And a good example of that are spores and larvae of the kinds of things that we work on, sessile benthic organisms, whose spores and larvae will often sense the chemical nature of a substrate before they make the decision to essentially glue themselves down for the rest of their lives in that spot. And the third area is chemical defense. And chemical defenses can be mounted against predators, either carnivores or herbivores, against biofoulers, things that would make you sick, and things that you're competing with, particularly for space. Now, in my lab, over the years, we've worked on almost all of the things that I just mentioned, really, except for chemical defenses against competitors. But the majority of our work, and particularly a majority of our work in Antarctica, has been on chemical defenses against predators. And so that's almost all I'm going to be talking about today. The chemistry in chemical ecology is usually because of compounds referred to as secondary metabolites, particularly in respect to the intentional forms of communication and chemical defenses. Now, calling something a secondary metabolite is a really clever way to distinguish it from a primary metabolite. Okay? A primary metabolite is something with a primary metabolic role, like a photosynthetic pigment or a respiratory enzyme. Secondary metabolites, on the other hand, normally don't have internal roles within an organism, and so are presumed to have ecological or otherwise external functions. All the work that I'm going to be talking about, as the title indicated, was done in Antarctica. We work at Palmer Station, which is on Anvers Island, here off the western side of the Antarctic Peninsula, directly south of South America. Palmer is very small. It's far and away the smallest of the three permanent U.S. research stations in Antarctica. It's got a maximum population of 45 people. About half of that can be scientists. But it's primarily a marine biological laboratory, and it's a really excellent facility to do marine biology-based research. It's on a small ice-free peninsula on Andros Island. If you walk up the glacier behind the station and look back down, there you see the ice-free peninsula, another ice-free peninsula next to it, and you see a few of the small ice-free islands that are within about a 3.5-kilometer radius of the station. And that's where we travel by small boat to collect organisms, to do experiments on back in the lab, or to do the few manipulative kinds of things that I'm going to be talking about today. Now, when we dive into these communities, we find ourselves in absolute forests dominated by macroalgae. And to put some numbers on that, I'll go back to some data I collected many years ago at Palmer. These are data from overstory video transects. It's like you flew over a forest in a plane with a video camera and then tried to figure out what was in the over the terrestrial environment, trying to figure out what you saw. You could tell a forest from a grassland. Within the forest, you could probably tell a oak tree from a pine tree, but you can't tell probably a loblolly pine from a longleaf pine, and you're sure not going to see the squirrels. So this is that level of data. I plotted percent cover as a function of the groups that could be distinguished on the videotapes of the day, and they tend to fall in, well, they fall into two main groups. The big bars are all brown macroalgae. If I did this in terms of biomass, this would be even a greater percentage of the flora. But there are a variety of red macroalgae. I'll show you pictures of many of these in a minute. There are not as many reds in terms of cover or biomass, but they're very important in terms of total species richness. There are a lot more species of red macroalgae than there are of brown. In the transects, the dots that you use to figure out what the percent cover is, none of them actually fell on an invertebrate that was big enough to see. But 11% was crusts or bare rock, but 70% or more actually of that crust and bare rock were crustose red macroalgae. So for a seaweed guy, this is a pretty good place, at least in terms of what's making up the community. In terms of biomass, 4.3 wet kilograms per meter squared might not mean much to you, but that's comparable to what you'd find in a giant kelp forest out in California. Okay? So never let anybody tell you there are forests in Antarctica. There are forests in Antarctica. There are forests of large brown macroalgae off the western side of the Antarctic Peninsula. And to show you what some of the more important algae look like, shallow waters down to, especially for Desmarestia anceps, 15, 20, even 30 meters at some site, will be dominated by one or both of these perennial non-acidic species of Desmarestia. Desmarestia menziesii is a little smaller and patchier and grows more shallow. Desmarestia anceps much bigger and much more widespread and goes deeper. They both look more or less like that, and these are two individuals, and they're big individuals of anceps, but they're not unusually big. And algae like this will just carpet the bottom with up to 100%, commonly 100% or nearly 100% cover of the bottom, over huge, huge swaths of the bottom. And it's that's where you get these kelp forest-like biomass levels. As you go deeper, at whatever depth at a particular site the Desmarestias thin out, they're replaced by other large brown algae, most commonly at least, by Hemanthothallus grandifolis. Again, this is a large individual, that's 8 meters, but they get 15, and they will also carpet the bottom like you see here, commonly covering 80 or 90% of the bottom. I mentioned red macroalgae. There are a lot of species. Again, they're not as important in terms of cover or biomass, but they're really important in terms of the diversity. Now, throughout the talk, I'm going to be talking about macroalgae, which is, I'll use the term seaweeds too. They're synonyms. Later in the talk, though, I'm going to start to differentiate the macroalgae by size. And when I do that, I'm going to start calling the larger macroalgae macrophytes to distinguish them from smaller filamentous macroalgae like you see here. Okay, so there's a lot of macroalgae in Antarctica, at least this part of Antarctica. So what? Well, we knew before our first field season on this project in 2000 that these algae were an important source of carbon for detrital food webs. And we knew that there were some animals that could eat them. but we didn't have an understanding of how much of all of that biomass was available directly to consumers. And so the very first simple question we asked was, are these macroalgae palatable to sympatric animals, the things that are there naturally and could eat them? Although there's a lot of biomass, there are not a lot of species, and so essentially we can work with the entire flora, or almost all of the flora, not a few. There are some very rare things, but we're able to work with pretty much the whole community. And if they're not palatable, we'd ask why. Well, to figure out whether something's palatable or not, you do a feeding bioassay. It's really complicated, but I got a technical diagram to help walk you through how they work. Okay? So here's a technical diagram. You take the potential food item and you offer it to a potential consumer and you say, does it eat it or doesn't it eat it? And you compare that to a known palatable control. Now, thallus is the phycological term we commonly use for what you'd call tissue in an animal. Okay? Now, I love this cartoon. It's even got a data sheet, right? But we're simply not interested in the palatability of mastodons, and so cavemen were not an ecologically relevant predator. Depending upon the specific question, we do a lot of work. I'm not talking about it today, but we do also work a lot with sessile invertebrates. We use a variety of local consumers. For today's talk, I'm primarily going to be talking about amphipods, but I will talk about a little bit of work with fish. So first question, is the fresh algal thallus material palatable? And then if not, why not? There's several reasons why something can be unpalatable. It could taste just fine, but be too tough to eat. Or it could be so low in nutritional value that it's simply not worth the consumer's time and energy to eat. And we do look at those things, and the toughness does play a role. I'm going to actually show you an example where we think toughness is playing a role. But by and large, what we found is that most of the unpalatability that we see is because of secondary metabolite defenses. And to figure that out, to get at that, what we do is a series of chemical extractions. We actually do two sets of solvents initially. One set of solvents that targets nonpolar solvents that targets lipophilic compounds. So we have a lipophilic extract, and then another set of solvents that targets the more hydrophilic components. So we have a second hydrophilic extract. And we take those extracts, dry off the solvents, and then add them to otherwise palatable artificial foods and do more feeding bioassays. So to give you an idea of what these look like, particularly with amphipods, these are some data from a former doctoral student, Craig Almack. And what Craig has plotted is the consumption rate of the fresh thallus material of 6 different macroalgal species to 2 very common macroalgal-associated amphipods, Gondogenea antarctica and Prostobingia gracilis. All of these bars that you see down here near the axis are not significantly different from 0. So none of those things were eaten. Gondogenea antarctica did eat Palmaria. That's the only thing that was significantly consumed here. Now, the reason Craig chose these 6 algae really doesn't matter for this talk, but I chose this as my example for 2 reasons. One is I'm going to talk more about Gondogenea and Prostobingia as we go through. Gondogenea is actually our main amphipod lab rat for these. But you're going to see a comparison between Gondo and Prostobingia. The other reason I chose it is because of these 2 species that I have in red, Placamium cartilagianum and Palmaria decipiens, which you're going to be hearing about more throughout the talk. For now, remember that Prostobingia gracilis didn't eat either one of these. Gondo did eat Palmaria. So the next question is, why not, and getting at the chemistry. I'm just going to show you the data for Prostobingia gracilis, which didn't eat any of these. So here are Craig's extract assays. Again, he's plotted consumption, this time of artificial foods. And the pairs of bars are the lipophilic control and lipophilic extract in green, and the hydrophilic control and hydrophilic extract in purple. And if we look at the 2 target species I told you I wanted to concentrate on, you can see that the lipophilic extract of Placamium was hardly eaten at all relative to the control. And so we can say then that the lipophilic chemistry can explain the unpalatability that we saw in the fresh thallus. Doesn't mean there's not something else going on, but the chemistry is enough to explain what's going on with the fresh thallus material. With Palmaria, neither of the extracts was eaten less than the controls, and in fact, that pound sign is actually to indicate that they preferred the lipophilic extract. You know, we were adding good-tasting things to the artificial foods. Palmaria, as it turns out, is one of the most widely palatable algae in this community, but we have found that Prostobingia and a few of the other amphipods don't eat it. It's clearly not because of the chemistry. We think there it's because their mouth parts just can't dig down into the tissue. But regardless, it doesn't eat it, but it's not eating it because of chemistry. The other 4 species, you can see that we could explain the unpalatability by chemistry in 3 of the species. For this one, Holopterus was another one where it was unpalatable, but the chemistry in this kind of assay can't be used to explain it. All right, so we've done a lot of this. And as I said, we've pretty much looked at everything in the flora that's at all common. And what we found is that most of the macroalgal species are unpalatable to the consumers that we use. And I've said at least half, but that's actually really conservative. It's a fair bit more than half of all of the species are unpalatable in a way that we can explain by chemical defenses. And that includes every single one of the big brown algae that dominate the communities in terms of biomass. And it also includes most, not all, Palmaria is pretty common, but most of the common red macroalgae. So looking back at these communities, the vast majority of that biomass is not, at least preferentially, going to be eaten by the local consumers. I compared this to kelp forests, temperate kelp forests in terms of biomass, but as many of you are probably aware, there's this paradigm about tritrophic interactions where the macroalgae, the kelps, are able to exist because their herbivores are controlled by the next trophic level. These guys are able to control their herbivores quite nicely all by themselves. There are a couple other unique things, or relatively unique things, about these communities. One is the enormous presence of amphipods, which for this crowd I'm sure you all realize are small crustaceans, shrimp-like animals. Amphipods are one of, if not the most species-rich group of animals in Antarctica, and they are incredibly common in these communities. To put some numbers on that, former doctoral student Yusheng Wang looked at 8 common macroalgal species at Palmer, and he quantitatively collected the amphipods on them and enumerated them. And he came up with number of amphipods per unit biomass. And we added that, we combined that with the biomass data that I'd collected back in the late 80s that I showed you, the same study that I showed you the cover histogram from, to come up with estimates of how many amphipods per meter squared of the bottom on these common algae that he sampled. They are estimates, but we have data from other places on the peninsula for some of the species that let us know that we're at least in the right ballpark. For Desmarestium enzizii, if you have a solid meter square cover of it, which it can do, it tends to be patchier than that, but you certainly get areas that are complete patches bigger than that. 300,000 amphipods per meter squared of the bottom. For Desmarestia anceps, which is the one that's far more widespread, 30,000 amphipods per meter squared of the bottom. And realize that that goes on for meter square after meter squared over huge swaths of the bottom. Not all of those amphipods are things that are likely able to eat algae. There are some suspension feeders and there are some predators, but most of them are. So there's a lot of things living on those seaweeds that ought to be able to eat algae. Well, another thing that came out of Yusheng's data, and we've got some other data on more recently, is the places where you find those ginormous densities of amphipods are on the things like the Desmarestias that we know they don't eat. On things like Palmaria, which I've showed you at least some of the amphipods can eat. They don't all eat it, but some of them can eat it. You find very few, at least during the day. Changes a little bit at night, but I won't get into that, but I can later if you want to know about it. Where this kind of pattern has been seen elsewhere, and people do see this other pattern in lots of places in the world, it's often attributed to the presence of omnivorous fish. And there are not a lot of fish in these communities. At least you don't see them very often. They're very cryptic. They hide down under the algae. You never see them up in the water column. But this species, which is far and away the most abundant, is an omnivore. It eats both algae and amphipods and other animals. I think it would prefer a steak, but it'll take a salad too. So a former postdoc, Jill Zamzow, wanted to test this idea to see if the presence of the omnivorous fish might be driving these distributions that we see. So she did some very simple experiments in buckets where she took Palmaria decipiens, this generally palatable alga, and Desmarestium zezii, which is the one that I showed had 300,000 amphipods on estimate. And she used the same two amphipods that I introduced you to before. So she used Prosopingia gracilis, which, as I showed you, doesn't eat Palmaria. It doesn't eat Desmarestia either. And in those cases, they always preferred to be on the chemically defended Desmarestia. When she did it with Gondogenea antarctica, which is the one I showed you does eat Palmaria, she did it in just normal filtered seawater, they preferred to be on, and they would be eating the Palmaria. A lot of you can probably see some amphipods on here. That's Gondo. This was one of these experiments where it was in clean water with Gondogenea. But if Jill took just a little bit of water that had been in a bucket that a fish had been in before, the pattern completely reversed, and they all skedaddled over and got on the chemically defended Desmarestia. This is one of those examples of an unintentional form of communication that I mentioned at the beginning. In larger scale experiments, Jill showed also that yes, indeed, an amphipod on the chemically defended Desmarestia is less likely to get eaten by the fish than an alga, or than an amphipod on Palmaria, and fish do like to eat Palmaria. So the amphipods, again, as people have seen other places in the world on chemically defended hosts, are benefiting from those hosts. Even though they're not eating the macrophytes, they are gaining an associational refuge there, and so are in a good place from that perspective. Another relatively unusual thing about these communities is that in the subtitle, you see almost no free-living filamentous algae, either as epiphytes or growing as turfs or growing epizoically. There are a couple very specific and relatively rare examples where you can find them, but by and large, you go diving in those communities, you don't see filamentous algae. Now, that's not because you can't be a filament in Antarctica. There are lots of free-living filamentous algae in the intertidal, primarily at Palmer at least, though, only in the fairly high intertidal and on fairly high-energy rocky areas where there's a lot of splash, so they're growing very high in the intertidal. Okay? Places where all those amphipods aren't likely to get, at least not get very much of the time. And a colleague, Akira Peters, who worked on King George Island, which is a couple degrees latitude further north, saw the same sort of thing. And we both thought, well, it's because of all those amphipods in the subtidal. You can't be a filament in the subtidal. You're going to get chowed down by an amphipod. Well, I decided to test that hypothesis. So I used an intertidal alga called Cladophora repens, which has a tufty morphology, which made it really amenable to this. And I just took tufts and I super glued them onto nylon washers, and I cable tied the nylon washers onto a shelf of a test tube rack and transplanted them out into the field. And I did this on very steep shores. Actually, around Palmer, most of the topography, underwater topography, is very steep. But I did it on steep shores, and I would put one rack on a buoy line 3 meters up off the bottom. The amphipods, during the day at least, which is when these experiments were done, are all down in the macroalgal canopy. And we never see them up in the water column. And we did some trapping just to make sure that they weren't all scurrying down and then coming back up in the water column. We weren't there to watch them. There are essentially no amphipods up in the water column. And by doing this on a steep shore, I was able to go not all that far laterally and put something at the same depth down in the canopy. And they were on buoys, but just up enough off the bottom enough that they weren't rubbing to abrade the algae. And I left them for about 6 hours, came back. Now, I did weigh them before and after, and all the statistics and stuff were done on the weights, but sometimes a picture's may not be worth 1,000 data points, but it's worth a few. And so just to show you the pictures, a couple of them I didn't get back to quick enough because after 6 hours, everything had been eaten on the bottom. The little bit of green you see there is what was in the super glue that the amphipods couldn't get at. This was the one that was eaten the least over those 6 hours, and even so, I think it's pretty obvious that these are smaller than these, and it was statistically significant in all cases. So even over 6 hours, there was a lot of consumption of these. So it is consistent with the idea that it's the amphipods that are excluding free-living filaments from the subtitle. So I've been careful to say free-living filamentous algae, because there are actually lots of filamentous algae in the subtitle, but they're all growing as endophytes inside the larger macroalgal hosts. And again, Akira had seen this as well and also thought, hey, they're hiding from the amphipods. And again, we wanted to look at that. And so one thing I did first was he worked further north on the peninsula, and I wanted to be sure that they were as abundant as they seemed to be. So I did a study of 13 of the more common, well, mostly common. There were 1 or 2 rare ones that I chose for specific reasons. But I surveyed 13 species of macrophytic algae. I subjectively scored the cover. I use the term cover even though they're inside just because it's convenient, of the algae. And what I found was in those 13 species, endophytes were very rare or absent in 5 of the 13. In the few that they were absent in then, I've since seen an occasional endophyte, so really I should probably just say that they're very rare. But in the other 8 species, they were extraordinarily common. In most cases, every single individual had endophytes. But there was a great range of cover. Okay? In some of the species, every individual had endophytes, but the cover would be fairly low. In one species, everyone had it, but I never saw one that I estimated as more than 3% cover. In some of the other species, though, there was a range. There'd be some with very low covers, but then a gradient up higher. And in one case, I had an individual that I estimated as 75% of the thallus was being underlain or covered by the endophytes. So for each individual that had endophytes, I took a piece of the thallus and I put it in culture under conditions that are really good for the growth of filamentous algae, but not good for the growth of a big piece of macrophyte. And after 3 months, this is what a lot of them looked like. There were both brown endophytes and green endophytes. And the brown endophytes would mostly, by that time, had grown up out of the thallus. The greens didn't, and so most of them got overgrown. But as you'll see, I was able to isolate some green algae as well. So from each of these, anything that looked different, I picked and put in unialgal culture, and I ended up with 99 brown algal strains. And then I had 2 green algal strains that looked very different, that fortunately had not been killed by all the brown algae that grew up. There's some morphological differences in the browns, but mostly they're just little fuzzy things. So to figure out what we had, Maggie Amsler, who's here, sequenced the ITS1 marker in all the brown algal strains. Juan Lopez Batista, who's a colleague who's over at Tuscaloosa and routinely works on greens and already was doing the greens, did this for us for the greens. And what Maggie found was those 99 unialgal brown species or strains represented 10 different species, and Juan was able to confirm that these 2 strains that looked different really were different. So then, knowing what I had other than just little brown fuzzy things, I took representatives of all of these species. In some cases, I took multiple representatives back to Antarctica, and I did feeding bioassays with Gondogene antarctica, what I told you was our favorite amphipod lab rat. And so I plotted the amphipod feeding rate on the vertical axis, and these are the 13 host species from nature. These are the filamentous algal endophytes from culture. Palmaria decipiens, the only one of these thallus fragments that actually gets much away from the bar, is that one that I showed you before and said that's one of the most palatable species, if not the most palatable species of macrophyte we find. These guys are candy. I did do statistics, but I didn't clutter the graph up with it. I'm pretty sure you'll believe that they eat those a lot faster than they eat the hosts, which made us suspect that, well, we know that they can grow up out, like the picture I just showed you, it made us suspect that, well, when they start to do that in nature, the amphipods are finding this candy and chowing it down. That's consistent with another project that Craig Almack, who I introduced you to before, former doctoral student, did. Craig collected pretty much all the common amphipods from the subtidal, pretty far away from the intertidal where there are filamentous algae, and he looked at gut contents and did some stable isotope stuff that I'll mention later. And he found that depending on the species, anywhere between 6 and 16% of their guts were filled with filamentous algae. So we aren't seeing filamentous algae in the subtidal, but the amphipods are finding them. He also looked at this experimentally. We're prohibited in the Antarctic from doing the kinds of studies that have been very powerfully used in temperate and tropical regions where you put poison out into the environment to kill the amphipods. I asked. Boy, did I get laughed down. But Craig did mesocosm experiments where he transplanted macrophytic hosts that had endophytes in them into tanks, some of which had natural densities of amphipods and some of which had no amphipods at all. He maintained those experiments for 6 weeks, and after 6 weeks without amphipods, not as dramatic as the 3 months from culture pictures that I showed you, but very much more individual macrophytes with emergent endophytes coming out of them, as we would have predicted. But far, far, far and away, the biggest difference between the mesocosms with and without amphipods was that the ones without amphipods were just covered with diatoms. Now again, for this audience, most of you probably know what diatoms are. They're microscopic algae. In the Southern Ocean, they are one of the very most important members of the phytoplankton. They're also very important in sea ice, and they're very important on the benthos. And this is just a SEM of a subantarctic alga showing it being very highly fouled with diatoms, which is generally thought of as a bad thing for a macrophytic alga because those amphipods have first crack at those diatoms have first crack at the nutrients, they have first crack at the light, and being fouled by diatoms is generally considered a bad thing for a macrophyte. So the amphipods are chowing down on these, and if you go back and look at Craig's gut contents, in almost every species of amphipod, the most abundant single category in their gut was diatoms. So it's very clear to us that not only are the amphipods benefiting from being on these chemically defended hosts, but the hosts are benefiting as well. So to summarize, so far we have these two really important groups, the physically dominant macrophytes, the numerically dominant component of the fauna, the amphipods, and I've told you that for the most part at least the amphipods are deterred from eating their hosts because of chemical defenses. Yet the hosts are providing a refuge from the amphipods, which benefits them. The amphipods are removing epiphytes, and I should have mentioned that that's certainly seen lots of places. It's seen in seagrass beds, it's seen on macrophytes otherwise. But these, again, are the two major components of the community, and I like to refer to this mutualistic relationship as a community-wide mutualism, at least with air quotes. And my title was Allies, Cheaters, and Thieves, and this is where the allies come from. I also told you, though, that we think that the incredibly high levels of amphipods on the chemically defended macrophytes are probably selecting for this common occurrence of endophytes, and that's because the endophytes are gaining a refuge from predation, the amphipods, by being in the macrophytes. But there's room for another arrow here, right? Filamentous algal endophytes can be completely benign, but they can also be pathogenic. As a matter of fact, in macroalgal mariculture, some of the most important, economically important pathogens are filamentous algal endophytes. Now, I've told you this wonderful story about this community-wide mutualism. Well, what if the amphipods are selecting for pathogens on their hosts, and so secondarily being of detriment? Does this still hold up? So a very recently graduated student Kate Schoenrock, looked at this for her master's and a significant part of her dissertation work. And what Kate did was to go back to that endophyte survey that I did, and she chose 4 of the 5 species of those 13 that had the widest range of endophytes. And we went out and collected them from sites where they were really common. She brought them back into the lab. This is actually one of her data slides. She used image analysis to get numbers for the endophyte load, much better than my subjective estimates from that I did in the survey. And then she transplanted those onto ropes that went onto concrete substrates that we put right back in the same community where they came from. And when she chose the algae, she chose ones with a range of endophytes from very low to very high. And she followed those for 6 or usually 9 weeks, depending upon the species, looking at their growth rate and their survival. And what she found was that in one of the species, the high loads of endophytes were clearly pathogenic. They were much less likely to survive, and they grew much more slowly. In 2 of the other species, there were statistically significant differences in growth or survival at the higher endophyte loads, but it was pretty mild. Biologically probably relevant, but not a huge deal. And in the other species, they were just completely, had no effect at all. So she was seeing in some of the species at the really high loads detrimental effects. We have all these other species that have low endophyte loads, and she really couldn't determine whether the low endophyte loads in these 4 species were having some effect or not, because there were almost none that had no endophytes in them. So she looked at a bunch of other things. She looked at 4 different photophysiological parameters. In only 1 of 7 species she did that with, was there any impact of the endophytes? And then it was very small, statistically significant, but small, and only in 1 of the 4 parameters she looked at. She looked at thallus strength in 2 different ways. 1 of 9 species she did a puncture assay, a penetrometer assay, if you're familiar with those. The thallus was weakened where there were endophytes. I said this was a tearing test, but I'm told that that's wrong. It's actually a breaking test. She also looked at 6 species whether just pulling them apart was easier to do if they had endophytes in them in the place where she was doing the ripping, and none of those were impacted. Another thing we thought might be going on, this is candy. I mean, it's growing inside some stuff that doesn't taste very good, but maybe it's worth taking a few bites of something that doesn't taste very good to get at the candy on the inside. And so we thought that maybe the endophytes were changing the palatability. And she actually did this in a couple different types of assays with 7 species and no effect at all. So she's looked at growth. She's looked at some physiological parameters. In the 1 species that I told you in her growth and survival experiments was clearly impacted at the higher levels, she went back out in nature, and she, from a bunch of different sites at 2 different times of year, collected lots of individuals and looked to see whether sporangial density was impacted by endophyte load. And there was no correlation at all between the level of endophyte load and either of the 2 types of sporangia that a red alga will make, cysticarps or tetrasporangia. So no impact on reproduction from at least the presence of reproductive structures. So getting back to our diagram here, yes, the endophytes can be detrimental to their host, but big picture-wise, we really don't think the few cases where we've been able to document that changes this overall picture. So one potential thing that could make this all fall apart, not an issue. But I started this whole bit of the story with this idea that the algae are chemically defended from their predators. Well, you probably know that there are lots of examples where there are animals that can eat chemically defended prey. Lots of moths specialize on chemically defended prey. Lots of amphipods specialize on chemically defended prey. And so if there were lots of amphipods in these communities that weren't just gaining refuge, but were actually eating their hosts, cheating this system, that'd be a problem for this story too. So we've looked for that, and we've only seen evidence of it in one amphipod species. There may be some really rare ones that do it too that we haven't looked at, but of all the common species, we only have evidence for that in one case. And that case is this guy, Paradexamine fissicata. And we got cued onto this from Craig Almack's, I talked about his gut content data, but he also did some stable carbon and nitrogen isotope signature stuff. And paradexamine had a stable carbon isotope signature that from his data set, which was fairly extensive in terms of the local macroalgae, could only be explained if they were eating one of 4 or 5 different red algae, all of which we know are chemically defended. And I also mentioned Yusheng Wang, who was a student who looked at the 8 common species of macroalgae to figure out their amphipod loads. And one of those species he looked at was this really common red Placamium cartilagianum. And paradexamine was over 2 orders of magnitude more common on Placamium than it was on any of the other algae that he looked at. And there weren't any disparities like that in the other amphipod species, and 30 or 30-some amphipods. So this suggested, hey, maybe paradexamine's eating this chemically defended alga, and this is one of the most highly defended algae for many of the consumers that we see. So again, to test that, Maggie went back and did feeding bioassays. And so she looked at feeding rate on fresh tissue of Placamium for paradexamine and 5 other common species of amphipods. And yes, indeed, the paradexamine was eating Placamium, and none of these other guys were. Again, none of those values are significantly different from 0. She did a bunch of other bioassays that I'll touch on in a little bit. But knowing this, she set up an experiment. So what Maggie did was to maintain paradexamine for 2 months on this chemically defended Placamium that we find it on in nature, and also on this very common, in some areas common, non-chemically defended Palmaria decipiens. Now, we don't think Palmaria is being eaten by paradexamine to much of an extent in nature because we don't find paradexamine on it, and the stable isotope signatures don't match at all, but it'll readily eat it if that's what you give it in the lab. So Maggie maintained these populations for 2 months, and then at the end of that 2 months, she took the ones that had been eating Placamium and she switched them over to Palmaria for a week. And that paradexamine eats really fast, clears its gut fast, and that's multiple passes of Palmaria through its gut to clean out any residual Placamium. And then she did 2 things. She gave some of the amphipods to Jackie von Salm, who is a doctoral student in my close collaborator Bill Baker's lab. I'll show you a picture of Bill in a little bit. Jackie and Bill are natural products chemists. They're the ones who do the chemistry in our chemical ecology. And Jackie looked at the chromatographic profiles of these amphipods. Now, to me, this is just a bunch of spiky lines. But to Jackie and Bill, those spiky lines from the animals that had been eating Placamium for most of the last 2 months indicated that they had the halogenated monoterpenes that we know, like anberine and epiplocoamine D, that we'd previously shown are deterrent to other amphipod species in their tissues. And when she did this with the with the Palmaria-fed paradexamine, those spiky lines said none of those halogenated monoterpenes. So these guys are thieves. They're stealing the chemical defenses from the algae. Or is it doing them any good? So the other thing Maggie did was to offer those amphipods to this most common omnivorous fish that we know is an important predator on amphipods. And she did the 2 groups of Palmaria. She also used a second species that is the most morphologically similar amphipod in the fauna. And here are the bioassay data. Percent eaten, almost all of the Placamium-fed paradexamine were rejected, and they were just spit out immediately. They weren't crushed. They weren't harmed. I mean, it was in their mouth and out. We train the fish that food comes on the end of long forceps, and so they'll try anything we give them that way. but they wouldn't keep the paradexamine-fed or the placamium-fed paradexamine in their mouth very long at all. That was significantly less than the ones that had been fed on palmaria for most of the last 2 months, and was also less than this morphologically similar alga, or amphipod. So not only are they cheaters, they're thieves, and the crime pays. They're able to get a benefit from stealing the defenses. This is seen in moths and frogs and some other things in the terrestrial environment. In the marine environment, the only examples of this where something is stealing chemical defenses and using them for their own defense up till this time was in apistobrite mollusks. So we were really excited about having an amphipod example of this. Paradexamine's a really good lab rat, and our new NSF project that will be going into the field next year, following up on this and using it to look at the costs and benefits of being able to do this kind of thievery is something we're going to be spending a lot of time on. Another thing we're going to be spending a lot of time on in our new work is the chemistry of placamium. I've been using the term Placamium cartilagianum because we don't have a better name for it, but we know that Placamium cartilagianum worldwide has an enormous amount of cryptic speciation, especially in the southern hemisphere. And we knew that this was different from, or at least some of this was different from what you see anyplace else in the world. We also know that other places in the world, Placamium cartilagianum has a lot of chemodiversity. They have lots of these secondary metabolites, like I showed, these halogenated monoterpenes, but within any population, different individuals will have different levels of the different monoterpenes. I mentioned Maggie did a bunch of other feeding bioassays. The paradexamine always ate placamium, but sometimes it ate it a lot faster than it did others. And we generally would collect the stuff from different sites, and it made us think, well, maybe there's some differences in the placamium. And maybe it's genetic, maybe it's something with the chemistry, or maybe it's some of both. So to get at that, we collected 3 closely spaced individuals from 7 different sites. Essentially, they all came from the same depth at the site, and they were usually within a couple meters of each other. And then Maggie sequenced 2 taxonomically informative genes, RBCL and COX-1, and what she found was we actually did have some genetic differentiation. There were 2 distinct genetic groups. These divergence numbers aren't enough that an algal taxonomist would call them different species, but clearly they're genetically distinct, so we just referred to them as phylogroups. And then we sent some to Ryan Young, who's a postdoc in Bill Baker's lab, and he did the same general kind of analysis that I showed you Jackie von Salm had done earlier, and he characterized the secondary metabolite diversity in each of these 21 individuals. And again, to me, this is just a bunch of spiky lines, but I can even tell that the pattern of those spiky lines are different. And when Ryan just looked at the chromatograms, he could put all 21 individuals into one of these 5 patterns. He didn't rely just on what his eye told him, though. He also did a couple statistical tests to compare them. And both those statistical tests gave you exactly the same 5 chemogroups. One of those is a Bray-Curtis similarity that you see here. And so the numbers and letters across the bottom here, the numbers are the site 1 through 7. The letters A, B, and C are the 3 individuals from the site. You can see these 5 chemogroups that fell out at 50% similarity are the same 5 that he saw, and he identified in some other tests. You see that the 2 phylogroups match perfectly. So there's a genetic component to that diversity. But there's also an apparent site-specific component. Most of you should be able to see the letters and numbers. So chemogroup 1 here has all the individuals from site 2, 2 of the individuals from site 1. The other individual from site 1 is over here in chemogroup 2. Same site, different chemogroup, but also genetically different. It's with 2 individuals from site 6. The other individual from site 6 is over here with the ones from site 4 in chemogroup 5. But again, different genetics. And these are the only 2 that were not the same as the others at their collection site, and in both cases, they were genetically different as well. So again, this is something that we're going to be spending a lot of time following up on to try and understand what's driving this chemodiversity. I hope I've convinced you that there are a lot of cool things going on in this system, even though I didn't spend any time on the sessile invertebrates that we work on too. We have this cool community-wide mutualism, as I like to call it, with the endophytes kind of adding to the intrigue. We have most of the guys in this community are alive in terms of the amphipods and the macrophytes, but there's that one cheater and thief that seems to be doing pretty well by being a thief. And those studies are leading us into some questions that we think are going to tell us a lot about other aspects of the costs and benefits and what's driving chemical defenses. So in closing, I mentioned Bill Baker a bunch of times. Jim McClintock was just down here earlier this year. He's also at UAB. He's the invertebrate zoologist, part of our team. We consider ourselves to be three completely co-equal, co-principal investigators on all the work that I've talked about. Also like to thank our field team members over the years, many, but far and away from all you see here. We get great support to work in Antarctica from the contractors who NSF contracts to do that work. And I always have to end by thanking my now, I'm at NSF as a rotating program officer right at the moment, but my predecessors who have allowed us to keep paying the bills all these years. And so I'll leave it there and be happy to answer questions.